Si, bonjour. So I'll have to speak in English because my French had not improved as much as I wanted. And today, like I said, I'll be talking about like rocky shores and hope that some of you get the idea that this type of environment is really good to ask basic ecological questions, okay? So there's some biology there holding for their life on the edge of the sea. The tide goes up and down, not like here, like 1.5 meters in Chile. Okay, so you have a pretty decent tidal range to work in. And the menu for the talk, so we stay together. First, I'm gonna talk about the good old days when David and I met. Then a little bit of what we learned initially. Then what we have been doing the past 30 years, and then a little bit to what we expect into the future. And I don't know if you see that image very well. That's in Coquimbo, in this town that David and Christophe went over in northern Chile. But count the number of species, and those are mostly rocky shore species. Okay, if you count very quickly and know how to tell them apart, there are like 30, and that's a good exercise anytime you go to a fish market anywhere around the world, right? So Chile is a long country, right? And it has the advantage that because it's very long and straight down, you can move very quickly through latitude. Okay, so the country is almost straight north-south. So every 100 kilometers, you move a degree of latitude. So if you go about 800 kilometers, you have been moving around 10 degrees of latitude, more or less, depending on how quickly you drive and depending on the roads, which is why I put that thing there in the corner, that twisted thing. Because back then, in the late 90s, there was no GPS, no Google Map, no nothing. And you had to rely on this sort of like a globe compartment guide to see where you were. And we had to design a sampling program. So we started like looking, okay, we want to go there and we don't know the road, we have to figure out the road. And here you're seeing sites that are spread between the two initial extremes of the sampling program, okay, between like 36 degrees and 29 degrees, sorry. And the one in the center is in 32, which is in Viña del Mar, where I live now, pretty much in front of Santiago. So the rocky shore ecology is rid of tomato.
You can proceed.
Thank you. So the rocky shore ecology, it has a very illustrious story, you know, like Robert Payne and that kind of people who kind of like set the base. Give you speaker, please. Okay, maybe you can put this one closer.
Okay, there you go.
J'ai coupé mon micro.
Okay. All right, here we go again. So the person here, Sergio Navarrete, back then actually hired David. We're talking 1997, okay? And he was just coming back from a PhD and a postdoc in Oregon and California with some of Bruce Mengi and Steve Gaines, like all the big names in ecology. And they had figured out a very simple way to look at intertidal communities that could actually be achieved in multiple locations. So we're in the age of ecology where people realize that if you want to study interaction, it's not enough to look at a single location, but you ideally need to go to multiple locations with multiple environments, things that now are pretty evident. But back then they were not evident at all. So we had this very like simple monitoring approach with that blue sponge on the edge. You'll see picture again. That blue sponge is to collect larvae. The little square is to collect larvae, also barnacles. And I draw a square there to give you an idea of how the world looks like, okay? So in the front corner of Ecology Letters last week, I think there was a picture of trees in a Mongolian forest. And if you have a lot of imagination, they look exactly the same. So it's a very good setting to study multiple species interacting with each other and that they don't run away from you too quickly. Okay. So you can look back at the same place over time, which is very important. Also, Chile, because it's so isolated, is not very diverse. So the species pool is kind of finite. It's not the Amazon, okay, which is very useful because you know the identity of your species. Okay, you can identify them quickly and keep track of them. Here you can see young Dr. Kaplan, okay, back in the 90s. So we sat down to work, okay, and do what rocky shore biologists do, and we started monitoring, looking at things over and over, the same places, counting species, writing them in a book. And I'll be talking about a paper that we just got accepted that says 2 decades, which is because we had lost the backup data for the earlier period, but we found it later. Okay, so you just believe me, the date in the logbook there, you can't read it because it's too small, but 1997, September 1997. So we're like getting there to a 3 decades. The thing in the tube on the corner, it's a thermometer that you can like take it out and in. Now we have a more sophisticated way of doing things, but we have been basically doing this exact same thing for 30 years in a number of locations. Okay, so we started a monitoring program in 1997, and we kept on doing it for about 3 years. Okay, this was before I left for the PhD, so we were doing it full time, just like counting, measuring, repeated every month, and then we got our first data. Okay, so this if the quality of the image here seems particularly bad, is because this paper was submitted on a fax. Okay, so that was the time when you had to submit like a thick envelope with like multiple copies of the thing, and then the queries of the reviewers by fax. So if you clip the PDF, that's what it looks like, and don't ask me for the data. But those circles there, the latitudinal gradient in coastal temperature was like taken out of these like paper maps and like written down in a paper. And when I made a table, when we made that graph, we were pretty proud of it. And the satellite image, you had to buy them in a CD that they will be mailed to you, and you had to use a very clumsy software to measure the temperature in the water. So it's like not like you have it now, which is great, but as technology progresses, we could calculate an upwelling index. Okay, how did we calculate it? Measuring the temperature differential between the coastline and the offshore on this very big pixel. But we could finally characterize the coastline in terms of the physics, okay? Exposed to upwelling, not exposed to upwelling, and we had a continuous way of doing it, which was pretty amazing. So this is, we were doing that in like year 2000 or something. And since we could characterize the site, we could sort of attribute what we were seeing in the community to the upwelling regime. So the shaded boxes are exposed or not exposed to upwelling according to our graph on your right. And keep that one in mind because it's gonna be improving with time, right? And what we could see is that not all sites were created equal, okay? Because when they teach you about like rocky shores and community ecology and so on, you would think of like the Mediterranean, that is like sort of similar, not exactly the same, but compositionally and functionally very similar. But as it turned out, like the sites were very different. Okay, I put the north and the south there, and you can, if you do a quick eye there, you can see that the north and the south are not identical at all. And there's a pretty interesting break here. Okay, I'll walk over. There's a pretty interesting break here that the mussels, which are like a dominant feature of the landscape, they suddenly disappear around a very fixed latitude, and some other species take over. Crustosalgi, they drop down there. In this paper, we chose to present functional groups because it was an easier way of presenting the data, but you will see that there's not a lot of redundancy. Okay, so there are not a lot of species per functional group, and the species are not very diverse in terms of having a lot of species within the genera. Okay, We have like as many genera as we have species, pretty much. And the thing is that across sites, the heterogeneity sort of coalesced into this very strong community-wide association. Okay, so if you correlated the amount of space occupied by one type of algae, it had to yield it to another type of algae. Consumers were negatively related to the abundance of the prey, and so on. So, for example, you have in the picture down there, you have a lot of low intertidal herbivores, and there's pretty much no fleshy algae. They ate it all, so they're negatively related to corticated algae, okay, and ephemeral. Okay, so in a nutshell, the community seems to be pretty strongly structured by interactions. Okay, the abundances of one thing, if one species goes down, the other one goes down. They're sharing space, and that's a very cool attribute of rocky shore ecology, that the interaction keep the community in balance, very, very orderly. So that's what we learned initially. Okay, this is like pure pattern. You go, you count, you do your correlation, and you end up with the general pattern. But as time goes by, now you can pay an artist and have like a very fancy view of what the Humboldt current looks like. Okay, and I'm showing you. I mean, if I may assume like everybody knows like upwelling and so on, but what you have in the center there, the South Pacific anticyclone, okay, which is the climate mode, so to speak, that regulates upwelling along shore in Chile. Okay, so we look a lot at what the anticyclone is doing more than measuring pixels, but this is like from a couple of years ago. And most of what I'm talking about is where the current branches have here. Here, that's Concepcion, where Christophe, I think, was for a little while. Okay, so as far as you can see, it's all one single upwelling zone. Okay, but I show you that already there's some heterogeneity in it. So now we had like better satellite images, but we were still like recycling the picture because it was still a pain to get the image. So different paper, same image, and different mistakes in the labeling of the site. Those are things that you see 20 years after. But now we could classify sites in terms of weak upwelling and strong upwelling. We can do a more fine job of characterizing them, and also looking at the variability in the satellite images and an upwelling index. Back then, we could measure every 3 degrees upwelling relaxation. Okay, so we could count the number of times that the upwelling index, switched from negative to positive. Okay, and we could see that around 30 degrees, all of a sudden, upwelling relaxations were becoming more frequent. Okay, this is because north of 30, upwelling is semi-permanent, and south of there, it becomes more seasonal as you move south, as you move forward. Okay, and this is due to the seasonal migration of the anticyclone. Okay, so we're getting a little bit better with the physics, and in terms of relating physics to what the ecological communities are doing, we could figure out now with more data over time. Remember that we were counting the larvae of the invertebrates that lived on the shore. So now we can see that there's a very strong linkage between the abundance of the adults, the percent cover, and the number of recruits that are reaching the shore. Okay, so we have a link between larval supply and adult abundance that is mediated by the upwelling regime, or so we thought, right? But that's what the evidence was showing us. And what we showed in this paper too, that I'm not gonna get that much into it, is that because the interactions, the ecological interactions are so strong, you do not see barnacles or mussels on the shore, even though the larvae are getting there, because the larval supply is too weak to keep the predators from removing them. So if you do your classical ecological experiment and you exclude the predators, all the time you will see that a community inside the cage that is similar to one that you see further south. But because the larval supply is not strong enough, the predators keep wiping up the colonizers. Okay, so it's a very neat example shown over by a geographic scale of community regulation. If you read your textbook ecology paper, this is like Joe Connell experiment with the barnacles in Scotland in the 60s. Same thing. But now you have proven that it works or geographic scale. And this was pretty much the first time anyone have shown that. Moving on, we wanted to see exactly how you partition that, okay? How you partition the role of the environment with what is happening in space, because things are spatially structured, okay? So it's not one big upwelling, non-upwelling, but things are spatially structured. So we did a shorter experiment, more focused. What you see, the time series here, this is temperature in the temperature logger partition on with a PCA, and you get this sort of like annual signal, okay, the strong seasonal signal, and you get a second PC mode, which we attribute to upwelling, and just using that, you could partition very neatly the sites that are north of 30 degrees and north of 30 degrees, and you get a couple that they have strong upwelling actually, and that they group with the other side. So you have an empirical way of separating sites in terms of their exposure to temperature variability that we're attributing to upwelling. And then you can use the same data and adult abundances plus the probable supply to partition what is structuring the community. And what you see here in the Venn diagram on the right, that temperature is very strongly regulating recruitment. It's accounting for about like half of recruitment variability, and space is accounting for quite a bit more. So in this very simple statistical model, you can partition variation between space, purely spatial processes that we're not accounting for them, larval supply and temperature, and you get this large unexplained variability, which basically we attribute to ecological processes that we were measuring in the other experiment, okay? So you have a way of experimentally determining what is structuring the community, and physical processes are important, their interaction around space, plus what species are doing to each other, and then you can predict community structure. So now we know how space is partitioned between species, okay? This is like pure ecology. But there's more to that, which is the identity of the species, because not all species are created equal, but we have this very limited diversity, right? And the area that we're studying is a biogeographic transitional zone. So as it turns out, around those 30 degrees of latitude, you have this, more than getting tropical species, the title of the talk is like tropicalization or borealization. There's no way we're gonna get tropicalization, because the next tropical species are like in the Galapagos up there, okay? And we're all the way down in the center because of the Humboldt upwelling, okay? So if you've been in Lima, the sea is fairly cold and you're at 14 degrees of latitude, right? So the effects of upwelling are very strong along South America, but you get a lot of subantarctic species that they have their limit of distribution around 30 degrees. So you have the ball kelp, the Lesonia kelp. There are two species that divide there. It's a chiton from the north, actually, that limit there, and the limpets in the picture. There are two species that overlap. So as it turns out, the biogeographic transition zone that is fairly narrow, it's like a 200-kilometer area, is very diverse, because it's the only part of the coastline where you find the species from the north together with the ones from the south, okay? And so for the ecology, it's a pretty interesting place because it's very species dense. And when you look at the environmental transition zone, more from an ecological than a biogeographic standpoint, it's very interesting because it's an environmental mosaic. So it's not just like two gradients that are meeting each other, but it's an area where the environmental regime switches back and forth. Okay, and the seasonal diagrams on the right, they show that basically sites from north to south are scrambled in terms of their temperature regime. Okay, so the one that is like Further down, the blue line is the second one to the north. Okay, so the coldest side is like almost the most equatorward side. And then they don't, basically temperature around the transition zone doesn't follow a north-south gradient. So now we're getting even more sophisticated. So now we can measure where the center of the South Pacific anticyclone is. And because we have been looking at the same area for a long time, you can go back retrospectively and see what the dynamics of the climate have been around that area. And as it turns out, the South Pacific anticyclone, as the tropic get warmer, is moving further south every year. And during some years, for example, El Nino year, it has this like spectacular southward excursion, and you get upwelling like at below 40 degrees of latitude, which you never do. So these are new things that are happening. And you can, if you remember the graph that I showed you below, now we have the upwelling relaxation every 1 degree of latitude, and they have been increasing through time. So upwelling further south is getting more variable. What we interpret from the data is that the upwelling events are getting more intense and shorter in duration or more unpredictable. I'll leave that to a physicist, but we are seeing an effect in the trends of chlorophyll in the satellite south of 30. So south of 30 is not that we don't have chlorophyll, it's just that the concentration near shore is getting lower because the water is getting swept offshore more frequently. And further up north, chlorophyll is increasing. Okay, and this is a paper from 2020 where we use data up to 2018, but we're just like redoing the analysis like 10 years later, and the trends are pretty robust. So around 30 degrees of latitude, you're getting a shift in the trend of the concentration. And the important thing is that with this change in upwelling regime along our transitional area, we are seeing an effect on the larval supply, in this case of 2 barnacle species. I'll talk more about that later, but at least for 2 barnacle species, you're getting divergent propagated supply trend. So southward, the trend is declining. They're getting less larvae. Up north, they're getting more larvae. The plots in the middle are just like a cool interpolation of all our sites in space. But if you pay attention to the one on the right, what you have there is a stock-recruit relationship. And the stock-recruit relationship is getting more shallow on the south because you're getting less and less larvae. But you have a storage effect. You still have adults because these adults are long-lived. They're producing larvae. And on the north, it's accelerating, okay? So as you get more larvae, your stock-recruit relationship is getting stronger. And this might be a presage of population collapse in the future. It's not that dramatic, but if you're a sessile species and you're getting less larvae, you might be doomed to extinction. Not to extinction because, sorry, these guys live between Peru and Punta Arenas, but the population dynamics are changing. And these are only 2 species, but I'll get back to that later. So now for the main dish, right? The plat du jour, which is basically how we think this community works, okay? So we were doing those quadrants, remember? And counting all the species and writing them in a little piece of paper. We're still doing it. And as time went by, this is what data looks like. And I'm gonna take advantage of the occasion that 2006 here, this is when I came back to Chile and we took over the northern part. These are collaborators down south that they were not very focused on what they were doing. So they were not doing their quadrats so often. Now we picked up again. But the spectacular thing is what I'll show you later. So in a nutshell, we have a lot of data on what is going on locally over these like almost 1,000 kilometers of coastline on the very same places where we're also measuring. Most of them were measuring the local temperature and the local larval supply, okay? At one time, I added up and the amount of area that we had sampled, it was like a little building. It was like I don't know, like 1,000 square meters or something, a lot of data. So this is the new paper. Mauricio is there in the water. This is like 3 days ago, and his dad just passed away. So I was like, say hello to him. This data is basically a rehash of the one that I showed you from 10 years before. Okay, but now the data is like really robust. So we went from having, like 50 quadrat per site to having like hundreds. So we're pretty confident that what you're seeing here, it's a reliable local pattern. And because now in the age of open science, you can go and download the data. I actually did the exercise and I downloaded my own data from my own paper and made an analysis that I'll show you later to hope that you're convinced. But what you see, these are invertebrates and these are algae. And you don't see any evident geographic gradient in diversity or composition. We have about 101 species. So it's not very diverse. 100 species is not a lot of species. We had 15 in the supermarket. Well, in the fishing market, right? But if you think our running joke was that there were more red algae in California than species in Chile in the Rocky Shore. And only in California. If you go to Oregon, there are already more. So it's not very diverse, which means that every species counts. And I'll show you why. There's somehow of a gradient poleward of algae, and that have been documented before because the species pool for the region is the South Antarctic. Okay, so there are several papers showing that we have a reverse latitudinal gradient for several groups, for crabs, for fishes, for algae, etc. So there's no evident geographic gradient, right? And so we had all this data, and I was thinking, like, how do we deal with all this data? Because you have the environment, you have the larval supply, you have the identities of the species, you know the functional traits of the species, you know their abundance, they're changing in time. And I was talking with another colleague called Paulina Hurto, and I had her picture, but I lost it in some of the iteration. And she was a postdoc with Otso Askainen. And I had just bought this book and I was like making my way through it very slowly. If you buy the book and try to go through it yourself, better go to Finland and take the class with Otso. That's my recommendation. You can do it, but but better to go take the class. So Mauricio took the plane and took the class. And we managed to go through the hierarchical modeling of species distribution, which is a very, there's a package in R, etc., but it's a very sophisticated way of partitioning how a species change in abundance or in presence absence together, given the taxonomy, the environment, their traits, spatial context, and so on. Okay, but basically what you learn once you wrap your head around it is their joint change, which there was no way that with my old school metrics I could do it. So the results were interesting, but when you go through them, first the temperature. Okay, so this is satellite temperature because we did not have temperature loggers in every single site, but they had been shown before. I mean, we were not the first ones to show it, that this is the mean and this is the anomaly, that basically temperature is not changing along the sector. So the only significant slope is this one in the mean, in one single site. And also the analysis for the paper is restricted for the years that we had data. Okay, so it's not every single year. Okay, and I'll show you the effect of including all years later. But basically, this is the number that's going on into the analysis. And we have no change in temperature, basically in the southern cluster and a little bit of it in the north, but the slopes are extremely shallow. Okay, when you look at temperature change, I don't know, in the Mediterranean, it's an order of magnitude larger. Okay, so things, if they're changing, they're not changing very quickly. So you're partitioning the effects of the different factors, right? You have tidal height, you have year, you have, I'm reading them here because I can't remember them all. And also like This is the annoying part of having reviewers that they don't believe you. We had a reviewer that wanted us to test the effect of season. And because we had so many samples, sure, we can do season, but of course, season is not important because there's so few species that we don't have a seasonally changing community. You might have like a bloom of green algae, but all the individuals are long-lived. There are few species that are always there. Okay, anyway, so season is not important. However, temperature seems to be important in the in the in the community weighted mean average. Okay, that's the metric here. For the black dots are algae, the open dots are invertebrate. So the mean temperature and the temperature anomaly with latitude, the bottom here is like north to south, seems to be important for algae. So algae are getting more packed together in the south as the water is getting colder, and apparently they don't like it to be so packed together. So they have a negative community weighted average. Okay. And with the anomaly, they're having a positive one, which to me is a little bit baffling. But the anomaly to the south are getting colder, right? But in a nutshell, you're not seeing a lot of change over the 30 years. This was interesting too, and again, was not very surprising because when you do this analysis, you use the phylogeny. But because there's so few species and it's so diverse, you have like all the algae and all the animal, we partition it by algae and animal, but phylogeny was not very informative. Okay, so it's not that some groups of species are changing and they are not, they're all reacting sort of in the same way. If you were seeing that, you will see here like big clusters of species from the dendrogram changing together. They are like all changing in the same way, except a few groups that are limited to some tidal height, but in nothing too exciting. I mean, species from the high intertidal, they don't live in the low intertidal, so they are high intertidal species. So evidently the tidal height is very important for them. And there I made a blog with the season, just so you get convinced that in taxonomy, the season is irrelevant, okay? Okay. This one was somehow more interesting to me, ecology, because he was saying that yes, time, the values are always this community mean weighted average, and if they are red or blue, it means that they're being positive or negative statistically. Okay, but it's a simplified way of putting it. So these are the random factors. So year is very important. They change in time. They're not identical year to year, because they're alive and they change. You know, it's not a major breakthrough, but time is important. For some few of them seem to be negative, most of them is positive, but they all react in the same way. Okay, and this is species with species. However, site seems to be very unimportant. So it's not very significant for any of the species, how things are changing in time. However, the platform, for invertebrates in particular, platform is basically the piece of rocky shore where you go and measure things, okay? And this makes sense because invertebrates, many of them, I put a little chiton there, they move around, so they tend to stick to the same microhabitat. So basically what you're seeing there is an effect of microhabitat. So your sites are all kind of like similar within in the site, but within the site, you might be seeing things different, which means that it's not trivial where you go and measure, right? If you're having, for example, a fixed sampling design, you might conclude that a species disappeared from there, but it's just like hiding under a rock, that type of thing. And the other thing that was interesting is that when you go back to the 2001 paper, okay, 20 years earlier, we did the whole school analysis with an analysis of variance where you partition variance, and we had basically the same conclusion because we did not have year, but most of the variance in the site was contained in the site. Okay, and the bench was not important in that analysis because in the bench you were not seeing the level of partitioning of variance that you see here when you look at every single species. Okay, so the species to species association, the advantage of the community weighted average, You were not seeing it in an earlier analysis, but you see it now. Okay, so your conclusion for the ecology that is going to multiple sites is that it's important which species you're looking and where you're looking at them. Okay, so if you're an ecologist and you have all this data and you have all this conclusion, you have to make an experiment, right? To kind of like test your idea. So I'm going to show you the experiment, but this is the final round. This is what I calculated downloading the data from the internet. I did a Ray Curtis. Okay, we all know the Ray Curtis dissimilarity, so it's similarity, and basically it shows that along the gradient, that's a confidence interval for the mid and the low shore, things have not changed a lot in time. Okay, just like looking at Ray Curtis and sparing yourself all the hierarchical distribution model. However, there's some sites that they seem to be getting more dissimilar in time, and they're the sites in the northernmost part, which are the ones that we know they get a stronger effect from El Nino, and these are the communities that I show you that are getting more larvae. So 2007, they were kind of barren, and 2023, they're all the sand, they're packed with mussels and barnacles. So there are some changes going on. The community structure is changing, but not that much, because I had a photo, there were too many photos, but 2026, like a few weeks ago, the same site, these are the same marker. Huh? These are the plate and the other thing, is the exact same piece of rock. The mussels were in retreat. So the community is kind of like switching back and forth, but it's not like changing very much. Okay, so for the experiment, this is Nelson Valdivia. Maybe some of you met him at some point. He did the PhD here in Bremerhaven, I think it was. So we were interested in how these communities are responding to change under this changing environmental regime. Okay, so we thought of we have done already the classical manipulative experiment of excluding the predator, etc. So now we thought, like, how about disturbance? Okay, we're gonna disturb this community. And how you disturb a community, you take a rock scraper and scrape the rock clean, and then see what goes on. But we did something. I'll show you like the results of like 3 or 4 different experiments, but they amount to removing the dominant species. Which are the dominant species? The ones that you see in the picture. It might be a barnacle, it might be an algae, or it might be a mussel. And they're like 3 species that we have identified as dominant, or that they're actually subdominants. Okay, so you scrape the rock, and then you measure through time what is going on, and then you reach until you reach an endpoint, or you run out of money, and you decide that is your endpoint. Okay, so and you see the trajectory of the recovery of the community, and you can get these different metrics of community stability. So we did this in southern Chile, Way Farm, still in the Humboldt region, of course, in around Valdivia. Called Valdivia, but there's a city in Chile called Valdivia. In Chile, it's a source of great confusion because Valdivia is in Valdivia. And not here up north, okay? So we did the experiment by removing the species. There you have like some pictures of a different experiment, but this is just so you get an idea. You get the control, and then after that, you follow the same one through time. You know, keep this full of muscle, the muscles are gone, they're on their way to recovery. So we did this for about 3 years. Visited every 3 months, okay? And then we measure how the function being space occupied by the different species, and composition being the composition of the original quadrant, okay? So you have replicated quadrant that they have the treatment, and you follow them through time, okay? And then calculate these like recovery metrics. And what is interesting here is that almost independent of the dominant species, once you remove the dominant species, the effect across the community is very similar. Okay, so if you remember, these are very species poor community. So the species dominating the community is very important in terms of what it's doing to the other species. okay? It's like providing habitat, providing shelter, providing space for recruitment, and so on. So across species, the recovery pattern was very similar, but it was not identical because algae, for example, recover more quickly. Okay, these are long-lived species, and some of these sites are recruitment limited. And in this particular experiment, we could not measure larval supply in all the sites because the sites down here in Valdivia are there are a lot of people that gather stuff on the shore. I'm not talking today. I'm avoiding actually talking about fishery or artisanal fisher, but the artisanal fishery in Chile have very strong views and idea, and if they see your experiment, they might think to take it out just because it's like it's their rock and not your rock. I don't know why it's their rock, but long story short, it was impossible to measure temperature in situ and recruitment in situ in the south. In the north, it's more remote and there are less people. You get to know them, but in the south, it was impossible. So we don't have all the elements of the equation. We don't have larval supply, and we don't have local temperature. That's why I have a satellite image there. You can always go back to the satellite. But the important thing is that, for example, in variability, when you were dominated by muscle, was very high. So it was very slow to recover because you need a larvae need to recruit, need to grow, need to form a muscle bed, and that takes time. It might take a longer time than the 3 years that we used. Okay, so not all the communities recovered back to where they were initially, but they were not really very different amongst them. Okay. Also, you can integrate the experiment in space. Okay, so remember that I'm comparing my quadrant over time, and then I'm pulling it within the site over time and seeing the trajectory. So when you pull the data across all the sites, okay, for all the sites across the region that were dominated by the algae, by the barnacle, or the mussel, you start seeing that this like variability start to dampen. Okay, which might not be very surprising, but we thought it was because your local conclusion might be really changed if you look into a bigger and bigger and bigger picture, even if you're going across this very large environmental gradient. Okay, so who's the dominant doesn't matter very much. So you can see, for example, that with the algae, it's like super variable locally, but then it dampens quickly. And the barnacles are very steady because they recover more quickly than the algae too. So if you think of the lack of change that we saw across the 30 years and the very quick recovery pattern, once you remove the dominant, you get this picture where we think, that there's an eco-evolutionary sort of mechanism behind this thing because it's a very species poor environment, but they're extremely strong in their interaction. Okay, it's like it's basically there are very few bricks holding the building, and there are no changes over the 30 years. And now the paper says 20, but you can trust me that now we fished out the earlier data, so that should be like 30 years pretty soon. Okay, so getting to the step. I'm just gonna show you a bunch of other things that we have learned in the later years. So the biogeographic transition. So as the studies mount and we have more and more study, we realize that it's a phylogeographic transition zone too. So most of the species that they go across the biogeographic boundary, they have some sort of phylogeographic change around that area. And this is way more noticeable, more detectable in species that they have limited dispersal. So your dispersal capability might be underlying this genetic population structure that you see. And this is for all kinds of species, for algae, for invertebrates, for fishes, and so on. So what we think in terms of the eco-evolutionary mechanism is that there's some sort of niche-based community structure. So not all species are able to persist or to establish biopopulations everywhere, but they can do it in many places. So you have this mosaic of interactions, okay? not all the sites are created equal, but they are made of this like reshuffling of a limited number of species. This again showing that there's a biogeographic break, but the connectivity matrix there is showing that oceanography, which is basically what it will drive dispersal of a pelagic larvae, it's also confounded. So larval supply is confounded with nutrient supply. Okay, so a place that might be bad for invertebrates might be good for algae, or vice versa. Okay, so we think there's something going on there with the environmental regime as we were been thinking all the way. So for a group of the sites, now we have close to 30 years of larval supply. This is like the raw data, we're still working on it, but every little bar is like one site, 1 month, okay, with like many replicates. So as I say, we're working through the data, but I'll show you later what we want to look with that. But the important thing is that the seasonality, remember the upwelling regime is changing. So what we want to look at now is if the seasonality, the tempo of the larval supply is changing. Okay, why? Because the north and the south have different larval supply regimes. Okay, they have different number of larvae and they have different seasonality. So part of this like niche-based difference might be in the temporal window where you get the larvae, and when you interact or you don't interact, or you colonize or you don't colonize. And also, this is a very old theoretical prediction that if you have a space-limited community and the larvae, the colonizers are getting offset, they're not synchronized, species can coexist. However, if they overlap, they might get synchronized and they might enter a limit cycle, and some of them will go extinct. That's a very old prediction that we want to check here. You remember the image of the satellite data for the upwelling through time. So if you look at all the data, okay, with this is the daily temperature product for the same site for the same time period, you get the exactly same spatial pattern. However, the slope is shifted, and it considerably shifted for the anomaly. South of 30, the trends are all negative at all the sites. Okay, so which is what we thought that we were not finding in the AVHRR data, but in the optimal interpolated data set, it pops up. Okay, so in the 2022 paper, we showed that the seasonality, this is the larval trend by month now, okay? The seasonality is strikingly different between the north and south. So not only the larvae are getting in different amounts, but are getting in different times of the year, which is not trivial if you live in the upwelling region, because the competition, for example, with algae can be more intense if you get there in the cold season when the algae are growing faster, for example. You might be excluded only because the time of the year that you get there. So we're following on the perturbation experiment. We already wrote the paper because we ran out of money with the other grant, but these experiments are simple, so you can follow them. And we set them up under different upwelling regimes, okay? So we have 2 in the north, 2 in the center, 2 in the south, that they have very different temperature trends. Okay, so we want to wait for a few more years to see what the perturbation turns into. Because if you give more time under different regimes, you might actually switch to a different state. And this is to wrap it up. So I went to a conference in July, and I think a good half or third of the conference was heat waves everywhere. This is a science paper with like heat waves everywhere. But I don't know, like we're like getting there with the data, but I don't see any heat wave there. We only see El Niño very clearly, El Niño and La Niña, which means that the heat wave as a global concern is not happening everywhere. I'm not saying that it will not happen here, but for us it's a very important conservation question, because if we're gonna plan, like future proofing and all that, we need to think if the heat waves are gonna happen or not. And as far as we have seen, I think it's difficult that we see one, at least during my lifetime. Maybe not, I don't know. I'm getting over the intertidal, right? Okay, so the digestive, that was not in the menu. So I hope you're convinced that the community structure is very robust, it's very predictable, and it has stayed stable for this sort of like 30 years now. We have a limited number of species, so very few bricks are holding the building again. Okay, so it means that you can reshuffle the species composition and get similar outcomes, and not all the sites are identical, but they're reliably different. Okay, we have an ongoing environmental change. I showed you already how the population dynamics are changing north and south of the transition, so we don't know what's going on yet. And as I say, I stay away of talking about humans, but humans are giving a workout to the environment, because in Chile, for example, is the largest exporter of wild kelp, of wild harvested wild kelp in the world. In all the world. Okay, it doesn't mean that it's that much, but these are small areas, and you see these are the same algae that we were counting. We had to like call it out because there was no algae. So there are the humans are having a very strong effect on this species poor environment, and I think we need to look at that. And also, as I show you, we have the phylogeographic break around the transitional area. So we think it should be an area of conservation focus too, because you have very rare genotypes that only live there. So the species is everywhere, but the genotypes from the transition zone are pretty unique. And also, it looks like an entrepot because all these species are overlapping there, and we have found already that some of them are hybridizing. So they're like two limpet species that only overlap there, but they are hybridizing. So it might be a source of new species, of actually evolutionary novelty for adaptation to this like changing environment. So thank you very much. And if you have any questions, I think we have some time to.
On peut peut-être commencer par les questions en ligne. Voilà, ça.
Well, I hope it was a good photo tour of Chile at least. You got to see a lot of sites.
Start us off. So one of the things that strikes me is it's true that there's this stability, even though you're showing that there's this changing environment. We're all predicting with climate change, there's going to be very large changes in community structure and everything like that. And you're not really seeing that in strong upwelling ecosystems. In South Africa, there's also, the southern part of South Africa being one part, there's also this kind of stability in community structure. And I've often had this theory that, the fact that there's this upwelling kind of gives a reset to the ecosystem every so often. It's bringing up deep water that's anchored and that's not affected by climate change. And that's sort of keeping the stability going. I'm wondering if have you ever tried like including sites in Argentina or something, or on the other side that's not in the upwelling system, and seeing if they have the same stability, or making sort of a regional comparison, because that would obviously break down on the other side because it's not an upwelling system.
Yeah, I'll love to do the experiment, but the non-upwelling rocky shore on the other side of South America is very species poor. And the guys in Uruguay, I think they're like getting there doing experiment, but there's no rocky shore school there, so to speak. No, it will be good, but most of the stuff that I've read, that I've talked with colleagues, they're like, if this species poor, like over there is like extremely species poor. And the sites that are not species poor that would be interesting are the upwelling areas in Brazil, in Cabo Frio and all that area. They have pretty persistent upwelling, and they are like amazing. You go there and it's like full of algae, different colors, completely different from the surrounding. Or in South Africa, you could go into Mozambique, that area. But the communities are extremely different. I mean, I've been there, and it's like that's why the geographic transitions are interesting too, because you get species from one side or the other side sort of replace each other. But for example, in California, which is kind of like the mirror of Chile, that is super species rich, you're getting like really big changes in community structure. I mean, they're published. I have read them, because the mussel beds that they have, they're disappearing in many places. And if you get the dominant species removed, like we show in the experiment, and permanently removed, you get like a very big change in community structure, and they don't have there any other comparable dominant species to take over. If you've never been in California, they have these Mytilus californianos that are like this size. And once you remove that, there's no comparable species to take over. And here, because all species are so small, you can take one out, and a sort of a similar dominant one will take over. So functionally, the ecosystem have very low redundancy. I mean, redundancy taxonomically, but functionally, maybe like they're doing the role of the other species. Any questions? Any fishes? No? I thought I should show some fishes. No?
I should mention that Bernardo is been on a sabbatical here in France. He's been here since last September. He's mostly been over at the university in but he's here until the end of February. So I think it's not bad media competition with them. So if I could ask another question. So the thing that's often striking me within Chile is this fact that you get, over very short scales, spatial scales, you get really strong changes in community structure, but the communities sort of repeat, as you said. And so I just want to make sure that I understood that was one of your results, is that within a site, you can get very different communities over short spatial scales, but then those different communities sort of repeat over very large spatial scales. So you have sort of a global homogeneity, but a local heterogeneity.
Exactly. And kind of a cool result is that the heterogeneity is structured in the mesoscale. So like over hundreds of kilometers, the site they will look like really similar, and then they will get scrambled. But they will be pretty much the same species unless you go over the biogeographic break, which will give you like a few different ones. But for example, like those big kelps that you see down there, until molecular ecology became common, we had no idea, and people had made all kinds of like counting cells and doing all the fine visual taxonomy, and nobody had realized that there were two species until we could sequence the algae that they're standing on. We thought it was the same all over Chile. There are three species, so there are more species than meet the eye. But visually and functionally, they're identical, the community. So even if it's a different species, you cannot like tell it apart. It's just like the rocky shore intertidal of the Humboldt Current. Okay. Thank you.